part out. This is like a little umbrella. It's kind of hard to see what it is, but these are things that are protecting us. And on this side, we have the adaptive immune system. We have two immune systems, really an adaptive immune system, which adapts itself to the actual pathogen that you are infected with. And then we have our innate immune system, which is with you from birth, and it takes care of any pathogen that gets into the body. Although it can't usually get rid of all of them, but they can hold them at bay until your adaptive immune system kicks in, which sometimes takes over a week to do. So that's what this is, like effector molecules, phagocytosis. All this part over here is your innate immunity, and you're born with it. It starts immediately upon a pathogen entering the body, and then this is your adaptive immune system, and that's what we'll talk about next week, next Wednesday. So it looks like, is there like an exam next Wednesday? I forgot.
Yeah.
So next Wednesday is actually exam. So again, we'll have the same situation. The exams are usually longer because the questions are a little bit more in depth for an exam than a quiz. So yes.
What chapter is the exam going to be on?
It will be from the previous exam. So yes, I don't know. I haven't looked at them yet.
Yeah.
So this one will definitely be on there. Yeah, this chapter will be, and then whatever we've had in the last couple chapters.
Quiz, what's that?
Oh, we're getting tested on the same chapters that we got tested on for the last exam?
No, 1013. That was just, okay, so like the stuff on the today's quiz, the 1013 was from the last exam, right? We never got quiz on it. So that's what she added to this quiz.
Oh.
But for the exam, it's just 8, 14, and 15 today.
So only 3 chapters.
Yes.
Yeah, I haven't even looked at it. But yeah, I'll just pick up the ones. And you know, it's really, there's a lot of stuff in the lectures, so there's plenty of 3 or plenty. Yeah, you guys.
This is exam we're doing on the 14th.
Is that a week from today?
2
Oh, 2 weeks from today. Apparently we have an exam 2 weeks from today?
Are you sure?
Just keep telling
I don't either.
I don't know.
I don't know.
The lecture
Yeah,
lab schedule says the 14th.
14?
So,
It says on the schedule.
Oh wait, that makes things different, guys.
So then we'll have apparently another chapter. We'll have a chapter that we lecture on next week.
Not next week.
Well, whatever it is, it's coming up. It's coming up. Whether it's a week from now or 2 weeks from now, it's coming up.
Is that the last one before the final?
I believe it is because I think, because we have June, something like June 6th is the last day of classes, something like that. And so this isn't even, we're just starting May. So, but probably it is because I think we have 4 quizzes and 4 exams. I don't remember now. I don't recall all of them. But anyway, it would be on the schedule. I put every single one of them on the schedule, so you'd be able to see. All right, so let's get back to talking about our immune system. Again, we have 2 immune systems. We have our innate immune system and our adaptive immune system. Our adaptive immune system is going to be next week. That's what we'll lecture on. So that would be nice if we get to put them all together. And so again, this little umbrella is saying that this is really what the immune system is doing. It's trying to protect you from pathogens. And this is really an interesting quote. Whatever that is. So this is an interesting quote. It says, and you have to think that this was spoken in 1908. It says, medical science today has set itself the task of attempting to prevent disease. And that is very true today, just as like it was in 1908. But it says, in order to achieve this aim, one must attempt, on the one hand, to find the disease germ and destroy it, which is what our body tries to do. And then on the other hand, to give the body strength to resist the attack. And so this was well spoken at the time because we know that if you allow your immune system to be able to take over, it can usually handle almost any pathogen, anything, COVID, anything else, it is strong enough to handle that. However, if there's any weakness in your immune system, then it needs help. So it needs something, some other, maybe antibiotic, it needs something to give it a little boost. But what you need to do then is only give it enough boost to let your immune system kick in. Because your immune system will your adaptive immune system will actually make antibodies, they'll make T cells, they'll make memory cells, they'll make a lot of cells that will help us in the future. But if we don't allow your immune system to kick in full strength, it will not ever make these memory cells, and it won't make antibodies, and it won't make the appropriate T cells. So we don't want to help the body too much. We just want to make sure that we step in before there's serious damage to the body. So this was good. This is Mechnikov. He's the one that discovered phagocytosis, and he got a Nobel Prize for that in 1908. So this is, again, he was a Russian zoologist. His name is Mechnikov. There's lots of different pronunciations of his name and spellings. He did a very interesting experiment. He took a thorn off of a rose bush, and he took this starfish larva that was clear. You could actually see within it with a microscope. And then when he stuck it in there, he noticed, and this is really hazy so you can't see it, there were a whole bunch of molecules that ran to, or swam to that area where the bacteria were coming off of the thorn. And so what was happening is these other molecules, these white blood cells, were actually coming to that site to destroy the bacteria that was coming off of the thorn. So he could see this firsthand, and what he did is he kind of drew what he saw. This is not his exact drawings, but he saw cells come, and I think we've kind of looked at this before. This is one of the white blood cells that he saw at the time. It's called a macrophage. This macrophage, when it's exposed to a pathogen that's on the outside, will reach out with what we call a philopodia. And a philopodia is a false foot. So its membrane extends out. And within the membrane, and I think we've seen something like this before, there's something called toll-like receptors here. So they're TLR, toll-like receptors. And they are within that philopodia that reaches out to the pathogen. What it's trying to do is trying to figure out, what is this? If it's a pathogen, it will bring it inside and it will destroy it. If it's one of our cells or something else that's unidentifiable, it would just be let go. So the toll-like receptors inside this philopodia will bring the pathogen in, and you can see we have these other little vacuoles in the macrophages, and they're filled with acidic material. So it brings it into the phagosome. That would be another vacuole here. And then it fuses the phagosome with this acidic material that is within the macrophage. And we're going to see it much better later on. So now that we have the pathogen in, and we fused it with this phagosome and lysosome, we are going to start to kill it. And when we kill it, it will just be dissolved in tiny pieces. And then what happens, the cell will take those tiny pieces, and it's mixed with things like pus, and it's extruded from the cell. But also it will take some of these little particles, and it will then put it on an MHC molecule, major histocompatibility molecule, that all of our cells have, and then it will display it on the surface. And then this macrophage will go to your lymph nodes all through your lymph in your body, and it will be searching for your adaptive immune system, your B cells and your T cells that recognize this pathogen. And if it can find your B cells then will take that in, it will recognize it as its pathogen that it can make antibodies against, and it will then start to make antibodies through a different series of events. So the idea is this macrophage will be able to display on its surface, on the MHC class 2 proteins, it will go to the lymph nodes, and then it will show the pathogen that has attacked the body to your innate immune system, and or your adaptive immune system. And when that happens, it starts activating your adaptive immune system. Okay, so it takes a while. So other molecules that can do this are macrophages and neutrophils can do this, and dendritic cells. There are a couple of other cells, but these are the ones that we consider phagocytic cells. They will take in pathogens. They take them and recognize them by toll-like receptors. Toll-like receptors will recognize things like maybe lipopolysaccharides on the surface, or glycoproteins or things that are on the surface that are commonly on the surface of bacteria. So even peptidoglycan it will recognize. So that means it's just general. It's not a specific cell. It's just any cell that has these particular things on it. So it's selective. It's not selectively killing. It's killing just because it happens to have something on its surface that this molecule recognizes. Okay. So anyway, this is phagocytosis. This is the their very first step that the body takes to try to get rid of a pathogen. Then the next step, when we think of the immunology, this is really what we're talking about. We have our innate immunity. This is a very quick response. We already saw we have some genetically encoded molecules already, dendritic cells, macrophages. They are with us when we are born. And then the goal of those type of cells is just to keep the pathogen in check until your adaptive immune system works. And again, your adaptive immune system is very slow to respond. You can see the macrophage had to display on the surface of the macrophage on the MHC class II. It had to go to the lymph nodes, which could take a long time. We have a lot of lymph nodes. Has to display this, try to find the B cell and the T cell that match that pathogen. And when it does, that can start to kick in. But even when that does, it could take another 4 or 5 days. So altogether, there's about a week or so that it takes to activate your adaptive immune system. However, it takes just seconds to activate your innate immune system. And that's the system that is trying to hold the pathogen at bay. Okay, and then once the adaptive immune system is activated, it makes long-term memory cells, it makes antibodies, it makes natural killer cells, it makes a whole bunch of cells that are going to be on the lookout for that particular pathogen. So another thing that we have various different cells that are going to be part of your innate system, so we'll see that on the next slide. But we want to look at the blood system because this is where your innate immune system is flowing through your body. It's going through the bloodstream. We'll kind of identify what's part of this bloodstream. So serum is part of it. We talk a lot about serum and what's in it. So it's a cell-free. There's usually not any cells in that. It's water, minerals, things like proteins, hormones, antibodies, etc. So when you have antibodies being made, they're going through the blood to maybe different places. So if you try to figure out if you've been exposed to something before, they can always take a blood sample, and they want to see if you have antibodies in your blood against whatever the pathogen is that they think you might have been exposed to. So anyway, this is serum, so you should know what serum is and what's contained in it. Then they have plasma. That's the serum, but these clotting agents also. And these clotting agents have 2 different substances in them. You don't have to know what they are. But what they do is they will then help form clots. So clotting agents plus the serum. And then just the rest of the blood, you can see like the vessel here, you have all kinds of stuff in there. So you have different cells like platelets, red blood cells, white blood cells, and we have a whole bunch of different white blood cells. But that's also going through the actual blood veins themselves. Okay, so this is just kind of an idea. We got 3 different ones that you should be able to recognize. Yes?
You said fibrinogen and prothrombin are the clotting agents, but we don't have to know them.
Yes, you don't have to know those names.
Okay.
Yeah, you just know that they're clotting agents. So we talked about fibrin and some of those things when we thought about what coagulase does and uses some of these proteins to actually coagulate cuts and wounds so that it can be hidden out in that clot and then get into the opening of the cell when the tissue when the macrophages have gone away. Okay, so these are a bunch of cells. There's only some of these cells that we really are going to focus on in the lecture. There's some up here like these basophils and eosinophils. These are ones that if you have allergies, etc., those are the ones that are releasing granulocytes, and give you all these allergy symptoms. We're not going to talk about those. We don't have time. Some of them will be mentioned in the very last lectures. There's about 5 lectures. It's called Diseases of the Body. those 5 lectures at the very end. They're somewhat short. Some of them are 17, 20 minutes long. Some of them are a little bit shorter. But we'll talk about them when we get a little bit closer. But some of these cells are talked about in those. So we will talk about the red blood cells. We'll talk about the platelets a little bit. Then we're going to talk about our neutrophils and our macrophages and our B cells and our T cells and our natural killer cells. So all of these cells we'll talk about, and also these dendritic cells. So those will be cells that are going to be important to us when we talk about how the adaptive and the innate immune system work together and how they get rid of the pathogens. So all of this is pretty interesting over here. It's good to kind of connect them to the different cells. And you can see that some of these are from the innate immunity, some are from the adaptive, and then we go back down here to this from the innate immunity. So these are on duty the day that you're born, and then these adaptive ones are come in once they have seen their pathogen. Okay. And so the circulatory system, that is how these molecules have to get around in the body. The ones that there's 6 of them, and these are leukocytes, and that first part of the word is leuko, it's white. So these are white blood cells, and there's 6 of them. These are the 6 that we had talked about over here. There's 6 of them, and so we want to talk a little bit more in depth about them. And there's just so many, 4,000 to 12,000, or 4,000 to 12,000 in just a microliter of blood. So when they're taking blood cell counts, all they need is a little bit of blood, and they can get a good idea of which white blood cells you're deficient in, which ones you have too many in, etc. Like your cat, and the neutrophils. So, yeah. So granulocytes, this is kind of what it sounds like for a cell. It has granules within the cell. And if it has granules within the cell, usually those granules are something to kill a pathogen with. So the cell will secrete these out, and then it will be able to damage the pathogen. So granulocytes, these are neutrophils, are granulocytes, and these are ones that usually just cause havoc with hay fever, etc. So allergic reactions. We are not going to talk about them. So the granulocyte that we're going to talk about is the neutrophil. It is a phagocyte, which means that it can actually gobble up the pathogen. So the pathogen comes into the body. It goes through that same scenario that we saw with the macrophage, when it has a philopodia, it reaches out, it grabs it, brings it in, and then it destroys it. So that's all part of phagocytosis. So this will be one cell that actually goes through phagocytosis. We're going to talk about the neutrophils a little bit more later because they're a very interesting cell. They only live a couple of days. So they're one of the first cells that are at the site of an infection. And what they do is they make this neutrophil extracellular trap. And this neutrophil will come in, it will spot the pathogen, and it will start exploding. So it's like an egg. If you broke an egg over a frying pan and you held it up and then you let it splat down into the frying pan, it gets to be quite large, whenever you splat down in the pan, and that's exactly what a neutrophil does. It explodes on getting in contact with a pathogen, and then it has a lot of granules that can start killing the pathogen, and not only one pathogen, that big splat usually gets a whole bunch of pathogens all at once. So it's doing its job to try to kill the pathogens, not going through phagocytosis, even though it can, but it can kill a lot more by splatting all over it. And then there's fibrin and lots of other tissues in there that will take all the pathogens that it has and clump them all together. And once it does that, it stops the pathogen from being able to do any damage to the body. And then the macrophages will come along, and then they will start chewing up this massive garbage that the neutrophil has created. And so the neutrophil is dead at this time, but then it has really captured a whole bunch of bacteria that cannot harm the body. It's stuck in this gooey mess, and then macrophages will come along and just clean that all up. Okay, so neutrophils do their part. Again, they only live a couple of days. They go through something called the neutrophil extracellular trap, and they can attack pathogens that way. So we're going to see this in action in a little couple slides away. So neutrophils, one thing, there are phagocytes. and they have granules that they can kill bacteria with. So other molecules, this goes to a 4, but we had 2 other ones that we are not talking about here. So here's 1, here's 2, 3, and now we start with 4. So these are monocytes. These are also phagocytic cells, meaning that they can gobble up another cell. They live a couple of weeks, but then they turn into these macrophages. They mature, and when they mature, what can happen is they can live for quite a while, maybe even up to a month. So I'm going to have you maybe close your computer. I have 3 people over here just staring at your computer. So can you close your computer, or can you sit in the front row so that nobody can be seeing your computer? Yeah, so all I see is the interest more in a computer, whatever's on your screen, than it is what I'm saying. Thank you. No, you're not. Yeah, people from behind you there can watch. So maybe just, keep it to yourself, whatever is on there. All right, so we have the monocytes. They are phagocytic also. We're going to have a little eye clicker coming up here asking you about the phagocytic cells, so you should be able to figure out which ones are. They eventually also could differentiate into these dendritic cells, and I showed you those. They look like starfish. And they reside in the spleen, the lymph nodes, and we'll see why this is important later on. This is a big filtering zone for pathogens, not even pathogens, molecules that come through the body. So this would be a good place for dendritic cells or other cells that can attack pathogens to reside. And then we have lymphocytes. So these are ones that are going to be the T lymphocytes and the B lymphocytes. These 2 are in the adaptive immune system. So our T cells and B cells, and they only increase once they have seen their pathogen. So the numbers of them will grow once they've been approved to be able to make either antibodies or whatever substance they're going to make to kill pathogens. And then we have something called a natural killer cell. This is also in the lymphocyte category. It's really an amazing molecule. It goes around and is looking for all of your the cells in your body, and it looks for specific molecules that are on it. So all the cells of your body have to be recognized as a cell that belongs to you in your body, otherwise it's going to be singled out and killed. So to do that, it has to make little housekeeping genes, maybe a gene that's like a common gene that's always for your heart muscle or your lung or something, but they have to make these all the time. They're always dividing, so it takes a lot of energy for a cell to do that. So natural killer cells go around and they're looking for cells that have good housekeeping, that they have the proper molecules on their surface. If they don't, what they do is they attach to them and then they will kill them. No matter why they don't have their housekeeping things on their surface, they will kill it. So this is indiscriminate, but they're looking for things that need to be destroyed because they're not properly maintaining their cell. And that usually means the cell has been infected with cancer. So if your cell is multiplying and dividing over and over and over and over very, very fast, it really doesn't have time to make all the kinds of molecules that need to be displayed on a surface, because it's just going to multiply and divide again and again and again. So those are usually the cells that your natural killer cell will recognize there's something wrong with the cell, and then it will automatically kill it. So when you think of that, cancer and also viruses, if a virus infects a cell, it usually commandeers the ribosome. The ribosome can't make the normal proteins and things that need to be put on the surface of the cell. And so a natural killer cell will come around and say, I don't know why you're not maintaining your cell surface, but we are going to kill you anyway. And so this is always good because it could prevent cancer from spreading really quick if a natural killer cell can prevent that. And they have shown that there are so many cancers that start early, but natural killer cells will knock it out before it gets to be enough inches spread. So natural killer cells are really important, and they go after virus and abnormal cells or cells that are perhaps have cancer in them. Abnormal just means that they're not maintaining the markers that should be on their cell. So something is wrong with the cell. And then we have the dendritic cells. These are the spiky looking things. They're on your skin, mucosal surfaces. Wherever there's entryways into the body, there's a lot of these dendritic cells are there. And they are also phagocytic cells. So if they see a pathogen, they can engulf that pathogen and destroy it. Okay, and all these come from your original cells that the body produces to start with, and then starts dividing into specialty cells. Okay, so for this, let's see. So here's a little clicker question that you can hold up and see what you've got. So which group of cells below are all phagocytic cells? So there's just a list of cells together, and you're looking for the one that. So these have to be all phagocytic cells. Okay, so I see some right answers and some wrong answers. Are basophils phagocytic? Remember, those are going to be your allergy cells, so they really don't do that. They're just going to secrete granules. So these are not part of it, so we'll get rid of those. And T cells are not phagocytic. They're part of your adaptive immune system. So then we can go to B. Macrophages definitely are. Natural killer cells, remember, they don't do anything. They just attach to another cell. They find out something's wrong with it. They don't phagocytose it. They just secrete something and they kill it. So they don't go through phagocytosis. So B isn't a good choice. And a T lymphocyte, B lymphocyte, again, these are lymphocytes. These are adaptive immune system. They don't take in things through phagocytosis. But these are the ones, these are the three that we really highlighted are the ones that are phagocytic cells. So these are the ones. Macrophages are really, really important. They go around chewing up things. The neutrophils, they splat on stuff. So they also can go through phagocytosis, but they do the most damage when they splat on stuff. Okay, so D is the best answer for that. Oh, too fast. All right, so the lymph system, you can barely see this is the body. So these are all the lymph nodes, if you could see them, all these little greenish color things. We just have, I mean, literally thousands of lymph nodes in the body. And I don't know if you've ever gotten sore throat or something like that, and you can really feel on the sides of your, kind of right by your ears and stuff, you can feel that you get swelling. And so your lymph nodes are working overtime, looking at all the pathogens, or maybe trying to help get rid of those pathogens, and a lot of that happens within the lymph nodes. So anyway, you have all these lymph nodes, and you have some primary lymphoid tissues, and these are where the thymus and the bone marrow is, and then we have the secondary ones where the spleen and the lymph nodes are. So I've kind of put these in color-coded for you so you can kind of see them up here. So the spleen has lots of natural killer cells and stuff. These are some of the ones that are filtering. Your spleen filters a lot of blood, and you have pathogens going through there all the time. And the lymph nodes, the same thing. These are where you circulate your macrophages, dendritic cells, B cells, and T cells. Okay, so these are the major things that you need to take home from this particular slide. And this is an actual lymph node, so you don't really have to draw that lymph node, but you do have to look up here. You can see that we have a B cell area up here. It looks like a candy corn, like those little candy things they have at Halloween time. So this little candy corn thing, this is where the B cells are, and then down here is where the T cells are. So, and this is the cortex area right in here, and down here is the medulla area. So you do need to know a little bit about where things are in the lymph nodes. So just know where the B cells and the T cells are going to be located. And then here is arteries for the blood, and then here's the lymph that comes through, and they circulate all through, up and down, up and down, up and down. And they get done with this lymph node, and then look at it, here's another one. Then they got to go to this one, and back and forth, back and forth. And then they go to this one, and this one, and this one, and this one, and this. So you can imagine how long it takes these macrophages that are displaying on their surface the pathogen that they have seen, and they want the lymph nodes to activate the B cells or the T cells. So B cells and T cells only have genetically the ability to to make antibodies against only one pathogen. And it's not a pathogen, it's a part of a pathogen. So that means any pathogen in the entire world, you have a B cell waiting for you to get that pathogen into your body. And the likelihood is just nil almost. You're going to see a few pathogens in your entire life, and the rest of your B cells are going to sit there forever and never see their pathogen. So only are they going to be activated when they actually are shown the pathogen that they genetically can make antibodies against. Okay, so your lymph nodes, they're in your neck, armpits grown, they're lots of different places, and they're exposed to those dendritic cells, and also the T and the B cells, that's where they're going to reside. And so also our memory cells for our B cells and plasma cells. And those will all make more sense when we have the next lecture when we talk about what B cells and T cells do. Okay, so when you get a pathogen in your body, there's a warning system that kind of goes out. Remember, you have macrophages that are going through your body all the time. You just have hundreds of thousands of them, and they're just circulating through the blood. So as soon as a pathogen is noticed by one of them, this is your innate immune system, it will recognize that there's something foreign in the body, and then it will start making an alert. And the way it recognizes it is there's something called pathogen-associated molecular patterns. And your innate immune system, its first job is to recognize the pathogen before any disease is occurring. So it has to have a very generalized way to recognize pathogens. There are only 10 toll-like receptors in the human body. I believe there might be 12 now, but I think 10. And it means that those toll-like receptors recognize only 10 different PAMPs, we call them, pathogen-associated molecular patterns. And that's enough of pattern for it to recognize almost any pathogen. So lipopolysaccharides, we know that they're on gram-negative bacteria. We see those things sticking on the top. Peptidoglycan, that's on almost all bacteria. Pili are on almost all bacteria. Double-stranded RNA, sounds like a weird thing, but a double-stranded RNA is in a cell. We never have double-stranded RNA. It must mean a virus is in the cell. So that's something that triggers your immune system to start being alert that there's a virus infecting the cell. And the same thing with double-stranded DNA. You don't have double-stranded DNA in your cytoplasm. It's either in the nucleus or the nucleoid, but this is our body trying to look at pathogens, and the pathogens would be the ones that had the nucleoid. So there's several others. There's about 10 different ones. So these are patterns that are general. So your innate immune system generally attacks molecules. It doesn't act specifically, it's all general. But that's enough to keep your body safe, because if something is in your body and it fits one of these categories, or about 10 different categories, it generally is tagged as a pathogen. So your innate immune system is right on the target, it's finding it, it's chewing it up, it's trying to stop any more from coming in. So it's going to hold this disease, or perhaps disease, at bay before it ever gets a strong enough reaction to cause disease within the body. So your neutrophils, dendritic cells, macrophages, when they see something, the first thing that they do is they display on their surface, and then they take off to the lymph nodes. But again, you have thousands and thousands of macrophages, so they communicate with each other. They send out signals. They're called cytokines, signals that are within the cytosol, and they send out these signals to tell other macrophages to come to the location. And then some of them take off and they go to the lymph nodes to show the lymph nodes that we are being attacked by this pathogen. And that will start your B cells and your T cells for inspecting every single pathogen that's brought to it on the MHC. So it will look at it and says, not mine, it goes to the next lymph node. No, it's not mine, it goes to the next lymph node. On and on and on, all throughout the body. Because there is only one B cell for every pathogen. Okay, and so it's somewhere in the body. All right, so the idea is that the signals are being sent out and you are getting more and more macrophages coming to that site. So you get inflammation, you get fluid buildup. The macrophages are coming out of the bloodstream. They have to go through a little hole in the bloodstream and some fluid also comes out. So you start getting swelling in your finger. That swelling pushes against some nerves in your finger or where else in your body, and you start having pain in that area. And redness. So this is really what inflammation is all about. But it's caused by the macrophages. So the same thing when we have COVID. We're in the lung. You have COVID in the lungs. So it sees a virus and it sends the alert. There's a virus in the lung. So it says, come to the lungs, macrophages. They have to go through the bloodstream. They wriggle out of the bloodstream. Some fluid comes into the lungs. Pretty soon your lungs are getting more and more filled with fluid because your macrophages are going crazy, trying to alert you that you have a disease and it's ending up in the lungs. And so that some of that is causing pneumonia and this buildup of fluids in the lungs. Okay, so interleukins, these are signals that are between leukocytes. So interleukins, so this is white, indicating white, and so these are signals between white blood cells. And so that's kind of the things that these macrophages, dendritic cells are doing. They're sending very specific cells to each other to tell what kind of pathogen, where it's located, what has to be done. And then chemokines, these are also special kinds of chemical signals that are being sent. They produce a gradient. So one macrophage says you need to come here as soon as possible. A whole bunch of a gradient is in front, and then it tapers off. So another macrophage coming in the direction, they go, oh, the signals are getting stronger, stronger, stronger. I'm going in the right direction. So chemokines are gradients of signals that are being sent out to try to attract other molecules to that site. So this is just really for information. I won't spend a lot of time on it. This is just tiny, tiny bits of types of communication that cells do. So this might be a single macrophage, and it has it sends out maybe cytokine signals. It sends out interleukins. It sends out these molecules that you're going to have for natural killer cells and things that will be just put onto the surface of cells. So all of these signals, and you don't have to memorize any of them, you just know that there's just a whole array of signals that are being sent out as soon as a macrophage or a dendritic cell, but we'll focus mainly on macrophages, send out signals. And chemotaxis is another thing. It just means chemical signaling. Kind of think of taxis as just like a little trail that's being left by all these signals. Okay, so that is just showing you all the okay. And here's another one. The signals that goes on in the body are just amazing. So here is just a cytokine receptor, and here's a cytokine coming in. So what does the cytokine do? Well, it comes in, it goes in this particular pathway, and it regulates things in almost everything. Every pathway you can see is coming to gene regulation. So what it's trying to do, it's trying to get to a cell to tell it what to do. And so the cell is figuring out what to do, and then it makes the proper gene to do the next step. So these signals are very specific to tell us either go through this pathway, this pathway, this pathway, go in and do this kind of thing to the genome, make this protein for us, make this, make that. So all of that signaling is going to produce some kind of gene that's going to be valuable for fighting that pathogen. So much is happening whenever a pathogen is entering. Okay, so again, nothing on this cell do you need to really, slide do you really need to focus on, but just the general concept of that signaling and stuff is so important for the body to fight a pathogen. So we're not going to talk about the adaptive immune system today, but I just want to kind of introduce what it is. Again, your adaptive immune system, it's not the first responder. It only becomes a first responder once it's been activated. So your adaptive immune system is your T cells and your B cells. Your B cells and your T cells have to acquire knowledge that a pathogen is in the body. And it gets that from the MHC class 1 protein or class 2 protein that a pathogen, when it's chewed up by the macrophage puts this on its MHC class 2 protein and then takes it to the lymph nodes. And that's where the T cells and the B cells are. So if it matches one of the B cells or T cells, then it will start to activate those cells. If not, it just has to go to the next lymph node. And that's why it can take so long to activate your adaptive immune system, because you can't find the right B cell and T cell to activate. So these take a lot longer than the innate. It could be several days, weeks, or usually not longer than 2 weeks, but at least several days to a week or so. And then once they are activated, you'll get antibodies made, you'll have memory cells made. Whenever that pathogen comes in your body again, then you will have a very quick response from your adaptive immunity, and it will be very specific for that pathogen. It will bypass any other pathogen in the body, but it will only go to the specific pathogen that it has antibodies or a recognition of. So it's very, very specific. Okay, so, oops, let me go back, maybe what this is. So here's another little quicker question. So which of the following is false?
Thank you.
So 1, interleukins are signaling molecules. Okay. How about C, primary lymphoid tissues are lymph nodes. Okay, leukocytes are white blood cells, and lymph is the fluid part of the blood. So it looks like this one, interleukin, signaling molecules, those are, right? Interleukins are, those are signaling molecules, and they're usually to white blood cells. And the lymph is the fluid part of the blood, we can see that. And, well, I wouldn't say it's the fluid part of the blood. The lymph isn't really in the blood, right? It's a separate system. It's a fluid that goes around all of your cells and it has nutrients and bathes them. So I think this is the one that probably we would not say is true. Okay, and then because your primary has your lymph nodes and then your leukocytes are white blood cells. Yes?
One of the slides says primary lymphoid tissues are thymus and bone marrow, and secondary lymphoid tissues are spleen and lymph nodes.
Okay, so you think this one also?
Yeah, it says on the slides that lymph nodes are secondary.
Okay. So maybe we have 2. But we agree with this one up here, signaling molecules, and this one down here. That makes sense? Okay. There we go. All right, so now we're getting back to the innate immune system again. I've talked a lot about macrophages. So you really have about 3 or 4 things that are going to be involved in your immune system. So the categories are cell-based, physical, chemical, or protein barriers. So if you look up here, it says cell-based barriers. These are some of the ones that we consider cellular ways to thwart pathogens attempt to get into the body. So your normal microbiota, remember, is very permissive or non-permissive. If it's non-permissive, it will not allow other pathogens to get onto the cells with it. So your normal microbiota will kind of outcompete a pathogen because most of your cells, your skin, everything in your body is just coated with your own microbiota. And only when pathogens enter and they can somehow get attached to your cells will they move those out of the way. And so you have your neutrophils, macrophages, and dendritic cells. Those are your ones that are going to be signaling to the adaptive immune system. And how do they signal to the adaptive immune system? What do they display? Yeah, they take in, they chop up the pathogen that they see. They take a little piece of the pathogen, they display it on their surface on these MHC class 2 proteins, and then they go around the body in the lymph nodes displaying this over and over. That's the only way that your adaptive immune system will be able to see that there's a pathogen. Okay, and then the phagocytosis that causes inflammation and fever, we know that things can flow into the body. So natural killer cells, these identify and kill virus and cancer cells. So these are everything that's called a cell base. So these you should be able to tick off a couple of those. And if you're asked cell base, maybe identify them or a short description of them. So these are cell based. Then we also have physical. I don't know why this is working very well. Okay, so well, this isn't. Okay, so let me see. No, too many to go. Okay, so I'm going to skip this one because I think this next slide is just, it's showing you how we have the microbiota is your first line of defense. Okay, so it's trying to make sure these other cells, and we've seen this slide before. Okay, so then we have physical barriers, and these physical barriers are things like your skin, so it's really hard to penetrate the skin for most bacteria unless you have cuts in your skin, so that's a physical barrier we have. The mucous membranes that you have on almost every opening in your body, it has a kind of a slick mucousy material so that pathogens can get stuck in it. These glycoproteins that are really what causes the mucus, these traps those pathogens or viruses in that material. And then eventually you get rid of that, you cough it up or you swallow it or something, and then the acids in the body will get rid of it. And then another way that we have to get rid of it is vomiting or diarrhea. This is something that we maybe do inappropriately by taking drugs to stop vomiting or diarrhea. If it is for some other cause, maybe yes, but if it is for a bacteria, what you really want to do is allow your body to get rid of it however it needs to do that. But it's very uncomfortable. Sometimes you go to maybe a foreign country, you get kind of a bug when you're there, and you want to take something to keep your enjoyment of your trip, but you really want to get rid of whatever pathogen you might have in your body. So allowing diarrhea and vomiting to occur is usually the best policy. And then tears and saliva, that's another thing. In tears and saliva, you have different molecules, and those molecules are getting rid of bacteria. These are also good to flush. And then urine, as you urinate, you're always flushing out things from the body. And then we have chemical barriers. These are effector molecules. So we have sweat glands, and those sweat glands make your skin more salty. So those will be safer for you. There's only a certain amount of bacteria can survive in high salt. We have the sebaceous glands, and those are kind of like glands that secrete this acidic fluids, and so that's also lowers the pH on areas. We have stomach acid, that's really a low pH. We have urine, that's a little bit acidic, and sometimes it can be basic also. And then you have the urinary tract for the women, where we have lactobacillus kind of in the urethra tract, and then what we do is we make sure that that is a low pH and keep any kind of pathogens and maybe yeast and things from forming. And protein barriers that we have. So lysozyme, I mentioned lysozyme. We also saw this when we had bacteriophage. They would land onto a bacteria cell, and then they would have to inject their DNA through the peptidoglycan layer. Peptidoglycan layer we know is put together with sugars, N-acetylglucosamine and N-acetylmyrmic acid. And so the bacteria would actually secrete lysozyme, and that would go through the cell, and then would start breaking up any of the sugar molecules. So lysozyme is something that we have in tears. We have them in a lot of different mucus and the sweat. So lysozyme is another thing, and that will destroy the glycosidic bonds that are in bacteria. There's another molecule that is a good inhibitor of bacteria. It's called transferrin. So the ferrin part means iron. It binds free iron. Many bacteria cells need iron as part of their enzymes in order to function. So if we can keep the free iron away from the bacteria by sequestering it with transferrin, that would keep the bacteria from being able to multiply and divide, because it needs iron in its enzymes. And the same thing with lactoferrin. This is found in saliva, tears, mucus, mother's milk, and it also binds free iron. Okay, so those two things. And then defensins, and there's many, many others, but these are just the ones that we want to kind of tick off. Defensins, this is what defensin looks like. It's cation, meaning it has positive charge on it. It's antimicrobial. It's a very small little peptide like this. And what it does is it gathers together, and then it will make a hole in the pathogen. So as it lands on the pathogen, it actually makes kind of a little hole in it, and then things can go in. So those big holes in the membrane of that pathogen will allow that to deflate, and more things can go in or more things can go out of the cell, and it will generally kill the bacterial cell. Okay, so these are just some of the protein barriers that we have. So it's almost amazing that we can ever get sick. We have a lot of things to keep us from getting sick. So protein complexes, this is the last thing that we're going to have in this particular category. These complexes are called, one of them is called complement. And if you take an immunology course, you could spend 2 weeks or 3 weeks just on complement alone. Complement is a group of about 30 proteins that are inactive. They're on a very long string of like a gene material, and then it's cut into little pieces to make a gene for each one of these 30 inactive proteins. So part of your genetic material, or the organism's genetic material, will have these complement proteins. We don't talk about any of the complement proteins, we just talk about them as a general term. So here's an example of what can happen to the complement. Here is just some of those 30 different proteins. You don't need to know any of them. And what they do is they will come together and they will land onto a pathogen, and they land side by side by side, and then they burrow a big hole, and now we have a big hole in the pathogen. So these complement proteins are usually triggered to go to a pathogen by like an antibody that has already landed on this, and it triggers the making of this this hole in the bacteria. So anyway, this is groups of complement, and we'll see this again later on. So what they do is they make holes in pathogens. And then interferons, the last one we're going to talk about for this. Interferon is another a molecule, and it's secreted by a virus-infected cell. So a cell in the body will get infected with a virus. It's too late for the cell to do anything about it. By the time it recognizes it, it already has the virus taking over its ribosomal functions, and it's not really even able to make proteins for itself anymore. But it does make one. It signals to actually make interferon. And so interferon is a protein. It's secreted from the virus-infected cell, the cell that's probably going to die, and it's sent out to all the neighboring cells. And it tells the neighboring cells that there is a virus, that I have gotten a virus, and if you get a virus that comes in the cell, do not replicate any of its genetic material. So it alerts cells around it to not actually take in genetic material from RNA from another virus. And there's codes on a piece of mRNA that the cell can tell that it's virus. So it will not produce any of those. So one cell will kind of sacrifice itself and send out this interferon to all the neighboring cells, and that will save them. So here's another thing, this is called a protein complexes. And again, many other responses like this, but these are just a few of the ones that you want to kind of focus on. All right, so which is not a good match? So lactoferrin suppresses iron, sebum, acidic secretion, PAMP, lipopolysaccharide, or complement degrades peptidoglycan. So which one is not a good match? Okay, so we're kind of all over the board. So let's go through the first one. Lactoferrin does sequester iron, right? So we have lactoferrin and we also have transferrin, okay? And sebum is the secretion that's kind of acidic on the skin, so that is helpful. And then PAMPs. What are the PAMPs are? This is a signaling, not a signaling, but this is a way to recognize another pathogen, and a lipopolysaccharide is one of the things that can be recognized on a pathogen. So pattern-associated molecular pattern, and this is one of them. And then complement doesn't degrade peptidoglycan. Remember, it actually just makes a big hole in the cell, but it doesn't really talk about that how it does that it's degrading peptidoglycan. So it doesn't really do that. It just actually burrows a big hole in the cell. So that would be the one that was not a good match. Okay. So you see we have a lot of different things that the cell has, and the cell tries to coordinate all of these abilities that it has to attack a pathogen. Because remember, it is really only trying to keep the pathogen at bay until your adaptive immune system can kick in. But that could take a week. And so in the meantime, your innate immune system is pulling together every single one of its sources that it has, and it's going to try to stop the pathogen. So this is the pathogen-associated molecular pattern that we just saw. These, again, are they're recognized what we call by toll-like receptors. They're on macrophages or any kind of a phagocytic cell. These receptors are the things that recognize the PAMPs themselves, and then that's going to trigger your immune system to take some kind of action. And macrophages, dendritic cells, neutrophils, they all have these PAMPs on them, or all have the toll-like receptors on them to recognize PAMPs. And these are what toll-like receptors look like. So this would be a receptor, and these are, there's 10 of them altogether as of this slide, but I think now they have discovered there's 2 more. But the important thing is that you can see this is, we have a specific PAMP for lipopolysaccharides, our specific toll-like receptor for lipopolysaccharides, flagellin, various membrane wall components, these are all these. And then we also have some that are on an endosome, because if things are brought in, you might have the DNA brought in of a organism, and you chew up the organism and the DNA is expressed, or as shown. So here you have double-stranded RNA, single-stranded RNA, single-stranded RNA. Again, another toll-like receptor, and double-stranded RNA. And these are things that when the bacteria is brought into a endosome, into a compartment, and the genetic material is exposed, these are toll-like receptors that signal to the cell that this is a virus, this is bacteria, this is something else. Okay, so these are all things. And what this is just showing is this little arrow means you will have an innate immune response, you will be making genes, you'll be doing stuff to try to get rid of those pathogens. Okay. All right, so from here, this is just a reminder of you. When you look at this, you can see that this is a gram-negative cell, and this is a gram-positive cell. So it's these things up here. Teichoic acid can also be recognized. Here's lipopolysaccharides. Up here, that lipid A, lipid polysaccharides. These are all things that are going to trigger a immune response. And this is the one I think I had told you before, that when a gram-negative cell is, you take antibiotics and it kills that gram-negative cell. The gram-negative cell starts to fall apart. When it falls apart, all these lipopolysaccharides that are up here on the top, they are going to spread out. And when that happens, your body thinks that it has a million bacteria that are now infecting it, but that's not the case. So this is, since it happens to be a PAMP, your immune system is going to think that it is a pathogen in the cell, and it's going to call more and more immune cells over to try to help kill it. Yes?
I actually have like 2 questions for you.
Oh, I'm sorry, I didn't see this part. Yes?
No, it's okay. For the slide before this,
Let me go back that way. If I can.
do you want us to know which, like, number of...
No. So just know there's something called a toll-like receptor, and that's what these are. And these are in the filopodia, and they are able to then look for PAMPs, which are pattern-associated molecular patterns. So these just look for these patterns, and these are the different kinds of patterns that they're looking for. And you don't even need to know all of them. There's just a couple of them. Just recognize them if you saw it on a question. Okay, but no number. You don't have to associate this number with this or this number with this.
Okay.
That makes sense? Okay, and so we'll just go on past this so we don't get stuck here. There we go. All right, so we're just going to go through these. This will end up our lecture, probably only a couple of minutes per each item, and then that will conclude the lecture. So your pathogen-associated molecular patterns, these molecules are going to trigger some response from the body. And these are some of the responses that can be triggered. Phagocytosis. That means bring the organism in, put them in some kind of a vacuole that has some acidic material and try to kill it. A net, so that means the neutrophil extracellular tract will make that net, and that will kill a whole bunch of bacteria. Inflammation and fever, your body will make a fever. If the temperature of the body gets higher, then some of the bacteria cannot live at that particular temperature, and so that will help. For inflammation and fever. It will trigger your natural killer cells, and we'll talk about what they do. These defensins, those are those little molecules that punch holes in cells, and then complement, and then interferons. We're going to talk about each of these separately. Oh, and then these are, this is just a stylized picture of the toll-like receptors on the surface, and they're looking for a PAMP. So they're looking for a particular thing that they can tell the cell, send it to the nucleus of the cell to tell it what we found. I don't know, maybe my battery is low. Oh, no, okay. So again, I'm not going to read these. You have these on the slides. You can read them over. We'll just actually start with the ones that we're going to talk about, so we don't have to go through all that. Here's phagocytosis. So this is an important slide. This will be a slide that you could actually be asked to draw, or you could be asked to fill in some blanks with it or do some labeling. It's really an important one, important discovery. This is where Metchnikoff got his Nobel Prize for this. So again, it's labeled on here, so I'll let you kind of read that on your own. But filopodia, remember, here's our extensions. It extends out from the cell. It has these toll-like receptors on the inside, and then it recognizes something on this particular bacteria. So the toll-like receptors are recognizing maybe lipopolysaccharides or something on that bacteria to say, this is a pathogen. So it brings it in, and this is a phagosome right here. So it brings it into a phagosome. That's where it brings almost everything. And then what it wants to do, it wants to kill it, so it brings it over to a phagosome. So that phagolysosome now is the phagosome and the lysosome, when they fuse, they call it a phagolysosome. And now this is the acidic material that's here, and it's going to start chewing up the pathogen that's in. So when it chews it up, there's little pieces of that pathogen that break up. And of course, those little pieces are going to be taken and put on the MHC class 1 or 2 of a bacteria cell. And of course, this is then going to take off and then go to the lymph nodes and try to display that to the adaptive immune system. And the rest of it, they'll just get rid of. So we all have had that. You have a cut or a sword and stuff, and you'll have some pus on it. And really, that's just old detritus from the bacteria. But these are really good descriptions. I'll let you kind of read each of them as you have time. And this is kind of a stylized thing that happens, but this is really, they send out these really long filopodia out there, and they feel the pathogens around, and they decide if those pathogens are something that needs to be in, or is that a human cell, or is that something else that's not considered a pathogen. Okay, but this is really an important slide that you should maybe make some tick marks by. Okay, so now we're going to talk about the neutrophil extracellular tracts. That's our second one. So again, here's a neutrophil. You can see it has these little granules, Those little granules, they kind of explode, and you can see it makes this netty material, and that's going to catch as many pathogens. The little red things in here would be the pathogens. It's caught a whole bunch of pathogens. And what it did is just keeps those pathogens from being able to get into our cells. So that's the purpose of your innate immunity, is to try to slow down the infection or slow down any bacteria that gets in. And the neutrophil definitely does its job. It's one of the first ones on site, only lives a couple of days, and it has these granules that it can actually secrete. So the neutrophils are first responder, and the PAMPs, once they figure out that there's PAMPs on here, then they start mixing or disintegrating, and they take all their nuclear material, etc., and they just make a big splot all over all the bacteria. So the plasma membrane breaks down, the extracellular fibers all are released, and all those granules are released with acidic material. So it eventually kills the pathogen, and then we just kind of wait. And so the macrophages will come along then and help clean this up, because these cells are already now dead. Okay, and there's platelets associated with this, thrombin, prothrombin, fibrin. All these things are just parts of the extracellular trap net that is put on. Okay. Inflammation is really important. This would be like a wound that you get, and when you get a wound, first thing is bacteria will come in. What happens is your neutrophils, remember they're the first ones on site. You have a lot of other things. These are mast cells, but we really won't talk about those. So we have macrophages, and your macrophages are sensing that there's pathogen coming into this wound. And what we'll do is we'll send out all these cytokines, sends out the cytokines that goes into the bloodstream, and that starts attracting a whole bunch more of the macrophages or other cells to come to that site. And every time they leave here, a little bit of fluid is coming out. And so that's where you're going to get this inflammation, the redness, soreness. So again, there's a whole series here. You can kind of do 1, 2, 3, 4, 5, 6, 7. It's a really good description of what happens with inflammation. And I think it's a good figure to explain what happens. All these are coming out of the blood. So you can see every time this sneaks through the blood vessel, it's going to have a little bit of fluid that comes with it, and when that happens, eventually the area will get filled up with a little extra fluid, and then that's where it will start being painful and red and pressing against the nerves to make pain. Okay, so this is kind of a good slide to check off. And the next one that we have, so fever, I mentioned that your body can spike a fever, and once it does that, it can make it so uncomfortable for bacteria that sometimes they will die, or at least they will not be able to multiply or divide very fast. And remember, that's the whole purpose of this, is to keep pathogens at bay, keep them from multiplying and dividing, so that eventually we can alert the adaptive immune system, and then they can come and start making antibodies or whatever they need to do to make to that pathogen. So low to moderate fever, that's usually what happens. If you get a really high fever, that endangers the body also. So fever-inducing molecules are called pyrogens. So that kind of sounds like a pyrographic or something, so for fireworks. But this idea is that this will cause the fever to be induced. So cytokines also will be inducing other cells to come to that location. Extracellular fragments from microbes like glycopolysaccharides, they will also induce inflammation and fever because, again, your body thinks it has a million gram-negative bacteria that are infecting it. So these lipopolysaccharides are very dangerous. They can cause a lot of problems. So the blood then is affected in the hypothalamus. It elevates the body temperature. That's a temperature regulator. And then your temperature can spike. And if it goes above 105, then obviously that is very dangerous. So the host can be affected also. So moderate fever is another defense mechanism the body has, but it can also backfire. Okay. So natural killer cells, these natural killer cells, they're in your body all the time. You're born with them. They're circulating all the time, and what they're looking for is housekeeping genes, and we'll go to the next slide and see that. They move in the circulation. They're looking for non-self cells. And it doesn't mean that it has to be a totally non-self cell. It just means a cell that is not putting the appropriate things on its surface to make it look like a self cell. So natural killer cells then will kill it. And it can be activated by interleukin 12, so it's something that can be secreted by maybe macrophages to say, hey, we think we have a cancer or we have something infected with a virus. You need to come over this area, and so interleukin 12 would be cause that. Also, interferon and other cytokines will actually trigger the natural killer cells to become more active. So let's look and see exactly what they do. So this also is a good slide to make sure you understand what's happening. So here is a cell, a regular cell, and it has this, what we call a common molecule on it, and it has an MHC class 1. All cells in the body have MHC class 1, only macrophages and phagocytic cells have MHC class 2. And there's a couple other kinds that also do. So you have a common molecule and you have MHC class 1, and this is a regular body cell. And then here's a natural killer cell. You see it has granules in it also. When those granules are triggered, then they will secrete them into the cell that they're attached to, and they will kill the cell. So what it's doing is it's just going around to every cell that it sees. If it sees a common molecule, it will take its killer activating receptor and it will attach to it. And this will trigger the cell to get ready to take secrete all of these granules into the cell. But it's waiting for another signal. So once this MHC class 1 is also recognized, then the killer inhibitory receptor will bind, and then that will signal do not disperse the granules. Okay, this is a normal cell. It will unlock and it will go to another cell. However, here's a cell that's virus infected. So this cell has not been able to put the MHC class 1 up on its surface. It has the common molecule, but it doesn't have the MHC class 1. So in this one, the common molecule, this connection, has sent a signal to the cell that says, unless we have another connection with an MHC class 1 protein, then we are going to disperse these granules. And that's it. This has not been noticed. So this cannot inhibit the activation of this signal that's already been sent. So that means that the cell that it's being attached to will be killed. So all these granules be released, and when they are, they make a hole in here. These are called perforin. So the hole comes in, and then granzymes go in the hole, and then they will chew up the cell. And usually what happens, and not always, but usually what happens, the reason a cell cannot take the time to make an MHC class 1 and put it on its surface is because it's either dealing with cancer, so it's replicating so fast it just can't do that. It doesn't have enough energy to be able to do that, or it's a virus infected, and so the virus is commandeered the ribosomes, and so the ribosomes no longer are making the molecules that need to, or the proteins for the molecules to go onto the surface. Either way, the cell should be killed. Okay? And so that's natural killer cells do that with no other instruction. They just, they're made to see if it doesn't have both of these on the surface, then it will kill them. Okay? So this one uses perforins to make holes and granzymes. These are, this is a protein and this is an enzyme. And they just consider these non-self cells. Okay, and I think this is going to be the last, two more, sorry. So this is complement. I mentioned how these are a series of proteins. There's 30 different ones. All you have to know is there's complement, not different ones. And here is a particular case where the complement now is landed on a cell. It's made a big hole in the cell, and so that cell is going to eventually die. So something has triggered complement to actually make a big hole in that cell to all 3 or 4 different complement proteins come together, and they make a hole in the cell. Okay. All right. And then this is, I'm pretty sure this is truly the last cell, the last one. I better not do that. This won't be able to come back. Sorry. Go back this way. So here's interferon, and I mentioned interferon was a molecule that when a cell gets infected with a virus, it recognizes that it's infected, and it recognizes that there's no way out. It's not going to be able to survive probably. So what it does is it sends in, once this recognizes that this is a double-stranded RNA that's in the cell, it recognizes that should be in there. So it assumes that it's being infected by a virus. So this is recognized by a toll-like receptor. That toll-like receptor sends a signal into the nucleus to say, we have a problem, we're infected by a virus. So that will then go to the chromosomes that are in there, the genetic material that's inside the cell, and then we'll actually start transcription of a gene called interferon. So once this transcription happens and this gene is made, it will be made into mRNA that will go outside the cell, that will be translated by the ribosomes. These interferon molecules will leave the surface of this cell and go to the neighboring cells, all the neighboring cells, and they all have these receptors for interferon on them. So the receptor then will take in the signal. It will send, the molecule does not go in the cell, but the receptor will take in the signal. The receptor end of the receptor will then send a signal back into the genetic material of its own cell, and it will say, hey, we have to go out here and we have to put antiviral proteins to make sure that our ribosomes don't make the proteins for this virus. Otherwise, we will then be also infected with the virus. So this allows one cell to stop the replication of viruses in all neighboring cells. And if you can stop the replication of virus, you basically stop the virus. So you'll have one cell that will be secreting virus outside, but when that gets into the neighboring cells, it's not going to be able to be replicated because it's already got antiviral proteins that are being made in all those neighboring cells. So a very clever thing that cells do. So interferon is something that protects cells from viral infection. Okay, so another good slide. Yes?
Is it only signaled in double-stranded RNA?
No, there's other ones. There's other toll-like receptors. Could be double-stranded. Could be any kind of thing that maybe a virus would have. And we have double-stranded, single-stranded, etc. Okay, does that make sense? All right, so we should be almost right at the end. I think we have this. Okay, we have this one and one other little thing, and then we're done. So let me go back here. So let's look at this. Which of the following are matched incorrectly? Okay, so the correct answer would be what we just covered is interferon is not for cancer cells, it is for virus cells. Remember, so interferon is going to be sent out when a cell recognizes that it has a virus inside, and it's going to try to send that interferon to the neighboring cells so that they will produce antiviral proteins ahead of time. And natural killer cell does have perforins that it secretes that makes the hole in the cell to kill it. Toll-like receptors and PAMPs, yes, toll-like receptors are going to be recognizing PAMPs, pathogen-associated molecular patterns. And lipopolysaccharides are endotoxins, so they're not exotoxins, they're endotoxins. Okay, so this slide is good also for, I mean, all the slides that we've had like this, these are kind of good questions that could be asked in the future, or things like this from a slide. Okay, so this is it then. I think we come to a little thing like this. Well, I mean, it might be helpful or it might not be helpful to you to kind of just go through here. So here's some physical barriers, here's some chemical barriers, here's some cellular barriers. So that may or may not be a good summary. Okay. So that is it. I thought it would take about an hour and 15 minutes, and it does. I tried not to go too fast, but almost all the information is right on the slide, so you can kind of review that. But the next one will be very similar to this one, will be an adaptive immune system. Thank you.
fucking